guest of the day, our BOG member, Truba Group of Institution. Good evening to all of you, and good morning at US. I hope. I, Dr. Rajesh Singh Pawar, welcome to all of you in this sixth interdisciplinary ethnoscience web conclave lecture series. Today is with us Dr. Lindsay Johnson as a chief guest from USA, and Dr. MS Sudhish from Amrita University, Kerala, and Dr. Nitin Swarnakar as a resource person and speaker. Here I would like to introduce about our guest, Dr. Lindsay. Dr. Lindsay Johnson joined us as a global technical marketing manager for pharma solution at BASF. Lindsay has 10 years of experience in solubilizing pharmaceutical ingredient and has published and patented a number of technologies during her time in academia and industries. So now I would like to request to Lindsay Johnson to focus on to the contribution of BASF towards pharmaceutical science and technology. Yes, Dr. Lindsay.
Yes, I'm going to.
Yes. Good morning or good afternoon, everyone. Thank you for having both myself and Nitin on the call today. We are very happy to be able to present to you from BASF. So I will start with a brief overview of BASF sort of broadly, but then really the heart of the conversation today is going to be presented by Nitin, Dr. Swarnakar, who will talk about enhanced solubilization of pharmaceutical actives through amorphous solid dispersions made through hot melt extrusion.
Your sound is breaking up a bit.
Yeah, yes. Yeah, Lindsay, your sound is breaking. Yeah.
Yes.
Is this better?
Can you speak again one more time? No.
Yes. No. No. So better.
Yeah, earlier it was okay. Now it is.
Is this okay?
I don't think it's kind of, it's maybe signal issue or something. It's breaking up a little bit. Now it's good. Now it's good.
Now it's good? Okay.
Yeah, perfect.
Okay. So we will talk today briefly about BASF, sort of broadly as a large chemical company. And then Dr. Swarnakar will present on enhanced solubilization through amorphous solid dispersions made by hot melt extrusion. Nitin, next slide.
Yes, I'm doing right now.
Okay.
Yeah.
I'm trying to see. Yep. Can you able to see now BASF purpose or something? Is it okay?
Share again. Yeah.
Oh my goodness. Okay. It
Yeah,
popped out.
Yeah.
There we go.
Yeah,
okay.
no.
So,
Okay.
perfect. Thank you.
Yeah.
So at BASF, we really see our purpose as creating chemistry for a sustainable future. That's important to us. We are the world's largest chemical company, and we really have a focus on orienting our practices from our supplies all the way to our customers to a sustainable mindset. Next slide.
Yeah, I'm trying to see it. Yep. Good.
Okay.
Yeah, already changed. I don't know whether you can see it or not.
There we are. So at BASF, our corporate commitment to sustainability covers every part of the value chain and the operations all the way to delivering to customers. So from a supplier standpoint, we source our raw materials responsibly. From an operations standpoint, we produce safely, and we also produce efficiently in order to decrease and minimize our impact on the environment in terms of CO2 emissions and other concerns. For customers, we help them drive sustainable solutions for their industries, and we value and treat all people with respect, whether they are our employees, our customers, or our partners. Next slide.
Is it?
So at BASF, we really want to be the leading chemical company for our customers, and we do that through the lens of innovation, sustainability, operations, digitalization, our portfolio, and our people, and always looking to our customers as the focal point of how we deliver and what we innovate on. So going one by one through each of these. Next slide. Our aim for innovation is to be the most attractive partner to our customers whenever they are confronted with challenges that can be solved with chemistry. For sustainability, we are successful in the long term. We believe that we are successful in the long term if our products, solutions, technologies all add value to the environment, society, and the economy. From an operational standpoint, we produce our products safely, efficiently, and reliably so that we can deliver products to all of our customers within spec and on time. We believe that digitalization is an integral part of our business because we believe that this is what creates value for our customers and will help us grow in the future our business and also improve our efficiency. For our portfolio, we are constantly focusing our portfolio and sharpening our capital allocation towards growth areas that facilitate sustainable delivery of new materials to global markets. And the last lens by which we serve our customers is through our employees, where we really rely on the engagement and the inclusion of our employees to be able to give them the tools and skills necessary to offer differentiated products to our customers through custom services and solutions. So BASF, we break down our activities across 3 segments. We have chemicals, materials, industrial solutions, surface technologies, nutrition and care, and agriculture solutions. We have sites globally for each of these segments, and we support activities in each of these segments with research sites, sales centers, Verbund sites, which are just very integrated manufacturing facilities, and R&D sites located across the globe. In the top left, you can see Tarrytown. This is just outside of New York City. That is where both Nitin and I work and are located. And you can also see on there the Mumbai site also pointed out. So this is just a snapshot of what we offer and how many sites we have globally. But you can see that we have a very expansive footprint. So specific to India and the region surrounding India, BASF has been in contact and in connection from a business standpoint with India since 1890. So across India, Bangladesh, Sri Lanka, and Pakistan, we have 3 wholly owned subsidiaries. We have a publicly listed company. We have a joint venture. We have 11 production sites, 34 sales offices, over 2,000 employees, and 1.5 billion euros in sales annually. So as you can see, this is a very strong footprint for BASF, and we're very happy to be in touch with India today. Next slide. One of the featured sites in India that we have, and this is where actually a lot of work that Dr. Swarnakar and I partner with, is an innovation campus in Mumbai. This was very recently opened. It was opened in 2017 and focuses on crop protection, process development, specialty chemicals, development activities that cover a wide range of industries. and just really to emphasize the growth that's occurring in India and how BASF values the opportunities there. Already in 2019, just 2 years later, we added to the site a creation center for automotive and appliance industry. Next slide.
Yep,
so all that, okay. So all that to say, BASF, you'll ask Nitin also, but I really love working at BASF. We offer job opportunities for everyone from students to professionals, and we really encourage you all to become part of our team and look at our website, basf.com/global/en/career/jobs, and I'll type the link into the chat here later. And this is where you'll find opportunities for you as students or as professionals to come join the BASF family. So just to talk quickly about or sort of prep for Nitin's discussion, so we both work in nutrition and care, which covers care chemicals and nutrition and health. This is going to be your shampoos, this is going to be your vitamins, this is going to be your pharmaceutical ingredients. All of that rolls under the business segment that we work in. Next slide. So in BASF, we think that there are some significant global trends that are influencing the nutrition and health market. Sustainability, aging population, urbanization, food safety, health and well-being, food security, and a growing middle class. These are all growing trends globally that steer where we invest and also help steer our portfolio, particularly in Pharma Solutions. So focusing on the customer's needs in Pharma Solutions, at BASF we deliver value-added functional solutions to formulation challenges, new formulations, new active ingredients, by leveraging our and Dr. Swarnakar's expertise in delivery systems and related technology. So he and I work in the solubilization platform. There are 5 platforms that are covered under Pharma Solutions in BASF. And he and I are in solubilization, which is where solutions with unmatched understandings of processing technologies enable you to achieve the necessary bioavailability of solid, liquid, and poorly soluble API. So with that, I would love to take this time now to introduce Nitin. He will be
Can you
presenting
hear
on...
Yes.
I can
you see the presentation?
Not anymore.
Yep. I cannot see myself also. I cannot see anybody. I don't know.
Oh, no.
Nitin, share you can share.
Yeah, I'm trying to see it. Like, how can I join? Something is going on here. I cannot see now. Link to join. Let me connect one more time so that maybe you can see better. Because I think maybe this is the main problem here. Like, you know, the internet is getting jammed because of so many participants we have. Perfect.
No.
Yep.
So your talk is over, Lindsay?
It is. I introduced Nitin now, and he presents on the topic.
Okay.
So, there we are.
Yeah. Before going to Nitin. Hello, Nitin. Do you listen me? Hello. Hello, Nitin.
I can hear you.
I think you
unmute
carry on with the
yourself.
program. I'll be joining you in the last session. Okay? Can you
No,
hear
sir.
me?
Just.
Okay.
Can you hear me now?
Yeah. Before going to start our the presentation of our resource person, Dr. Nitin, I would like to just request to our chairman, Sri Shyam Rathorji, who is connected with us, to say a few words about the event as well as our guest. Welcome, sir.
Yeah.
Okay. Thanks a lot, Dr. Rajesh. I welcome Dr. Lindsay, Dr. Nitin, Dr. Sudesh, and all other participants to this event. And I'm very grateful to have the knowledge sharing with all of you. And I especially thank you all for giving your time and support. And it will be really helpful for the students and the participants to get a good knowledge. And thank you very much for making this event successful. Thanks a lot and giving me a chance to welcome you all. Once again, welcome you all to this.
Thank you, sir. Thank you for your kind words. Now I would like to introduce our resource person, Dr. Nitin Swarnakar. Nitin Swarnakar is a scientist, Global Technical Marketing Pharma Solutions, Tarrytown, United States. His background and education, he has 13 years of combined as academic institutions and research and industry experience. He had finished his B.Pharm, M.Pharm and B.Pharm and M.Pharm from Dr. H.S. Gar University, Sagar, and Doctor of Philosophy from Naipur, Mohali, India. The experiences of Dr. Nitin, currently he is associated with BASF, and he is taking care about solubilization platform since 2019. He is also working with the TISO RX Pharma, Ponemo, California. He's earned postdoctoral researcher in various universities and 11 Indian publications and 4 US patents and more than 30 publications around the research in the research areas. His key formulation specialist, solubilization of small molecules and peptides, lipid-based formulation including liquid crystalline, nanoparticles, liposomes, capsules and seeds. Seeds means self-emulsifying drug delivery systems, polymer-based formulations, microspheres and PLGA nanoparticles. So with this, I would like to request Dr. Nitin to start their presentation and start their presentation on to the topic, which is enhanced solubilization through amorphous solid dispersion via hot melt extrusion process or method. Meanwhile, I would like to introduce our another panelist, Dr. Akash, who is from BITS Plani, Hyderabad. Welcome, Akash.
Thank you, Professor Pawar. Thank you.
So, Nitin, you can start your presentation. Yeah, Nitin, unmute yourself, please. Unmute yourself.
Yes. Now can you hear?
Yeah.
Thank you, Dr. Pawar. Thank you, Lindsay. Thank you, panelists and dear audience. Today we're going to discuss about enhanced solubilization through amorphous solid dispersion via hot melt extrusion. So what we're going to discuss here is, first we'll discuss about why we need solubilization, and then we're going to discuss about amorphous solid dispersion technology, in which we're going to cover about different type of amorphous solid dispersion we generally use and what are the problems we generally face, and then we're going to discuss about hot melt extrusion, which will touch base with the basic aspect of hot melt extrusion, how to develop amorphous solid dispersion, and what are the parameters we should have to look into during the manufacturing the product. And then we're going to go over the conclusion part. Can you see the presentation right now?
Yeah.
Okay. So just a minute, I need to adjust my screen first. Okay. So why, first of all, let's discuss why we need solubilization. Answer is very simple. Drug which not dissolve, cannot get absorbed, and will not be therapeutically effective. So meaning the drug is poorly soluble in biological fluid or difficult to absorb. The reason for this poor solubility is either because of the crystal lattice is too strong. It means that drug cannot be converted into the molecular form, and it cannot interact with the biological membrane, or it is too lipophilic. So we can say that if the drug is too lipophilic, the water cannot interact with the molecule. And hence, the two broad category we can see in overall drug molecules. So in the layman term, we can say brick kind of molecule, which has strong crystal lattice and high melting point. And we can say one more is the butter kind of molecule, which is having high hydrophobicity and low melting point. So these are the broad category in terms of layman language. And in terms of the scientific language, we can say these are BCS class 2 and BCS class 4 molecule, which has solubility and absorption issue. And if we compare that, the current marketed 2020 molecules with 2010, and we can see the bar diagram in this presentation here, which I was trying to point it out here. So overall, the molecule has increased 70%, meaning the formulator are facing more and more challenges. In order to find suitable technology, scientists have proposed one kind of graph which you can see here: solubility versus hydrophobicity. Solubility means log S, and hydrophobicity means log P. And they try to understand like what kind of technology going to work on this molecule. So this is like plotted like 6,000 molecule, and we also try to map it out what kind of technology is more useful for what kind of molecules. But ultimately, there is no such solution. We cannot decide based on the drug property alone that which technology is more suitable. Sometimes company has their own expertise. Sometimes like one company has expertise in the lipid-based delivery system. They will not try to establish something which is very novel for them, and they need to establish new technology in the R&D. So this is main drawback right now if we look into this graphical representation. So overall, we can say there is no one-size-fits-all solution to the solubility, and this is a major problem. So BASF generally have expertise and excipient through which we can generally develop technology which is more practical oriented and consumer friendly. So this is a major overall area of BASF where we have excipient and expertise also. And if you look in the next slide, these are the main trend industries right now going on. If you look across the overall platform, we have these kind of three pillars we generally focus, and especially like we have more specialty area also where we focus. But at this moment, this is like main pillar of the BASF. So if you look, we have amorphous solid dispersion pillar, we have lipid-based delivery system, and then we have liquid solubilizer and parental. So in liquid solubilizer, which is like a more micellar kind of system, it covers parental molecules. And if you look the lipid-based delivery system, we have drug solubilized in the lipid droplets and stabilized by the surfactant. So this is a major kind of system which we have discussed. I think maybe a study, if you get the chance to discuss about LBDDS, we discussed yesterday in one webinar. And today we're going to focus on the amorphous solid dispersion, which is another kind of technology in which drug is molecularly dispersed, or different kind of drug is dispersed into the polymer. We'll discuss in detail. BASF key excipient for this kind of technology, I'll touch base only amorphous solid dispersion. We have Colidon VA64 and Soluplus. We're going to hear this kind of excipient more and more once we talk about ASD formulation. One more important feature of amorphous solid dispersion, for those who don't know, like students, what is this kind of technology? So in the life, we generally see like two kind of candy. One is simple candy, which is like hard, which is like a molecule. And if you see the cotton candy, which is like a amorphous solid dispersion, this is the same technology which we can say we can correlate candy versus cotton candy. So cotton candy is big, You can feel it. It's really soluble in mouth. You put in mouth, it immediately it cannot dissolve. So that technology correlates with this amorphous solid dispersion. So for students, this is a kind of correlation we can see. Now coming on to the amorphous solid dispersion. So first of all, what is amorphous solid dispersion? It is much like a sugar dissolving in your coffee. It's very simple. So you take sugar and dissolve in a coffee. That's your, or maybe we can say in India it's tea, which I've changed to coffee to tea because people prefer tea. So this is the same like dissolving the solid in liquid. And if you evaporate that liquid, it's going to convert into the solid solution. So if you look this tablet, it is a kind of having a solid solution. And if you zoom it, and when this tablet will disperse into the biological fluid, overall polymer will orient in such a way that the hydrophilic site will move toward the fluid and lipophilic site will become close together to form a one core. And you can see the drug molecule. It is already trapped inside this lipophilic region. And this is how the ASD looks like in biological fluid. So you can see hydrophilic element we have, lipophilic element, and drug complexation. So this is very simple. Sometimes what people generally do is, in order to make more dissolution, they include plasticizer, sometimes solubilizer, that we're going to discuss in today's meeting. So pretty much this is kind of polymer you can see, and we have two polymer which I was trying to say is BH64 and Soluplus. Now coming on to what happens like if you prepare a solid dispersion and what advantage we generally get. So in case of limited dissolution rate, decreasing the particle size of the drug crystal can improve the solubility. This is a major thing. If you can see here, drug are in crystalline form. When you give energy, they're going to orient in different kind of assembly. It can be amorphous. And then when you give more energy to them, they're going to become more molecular form. So as you can see in this aspect, API surface area is increasing. They are becoming more distorted. And because of increasing API surface area, now another inherent problem which generally comes is thermodynamic stability, which we can see here is decreasing. So thermodynamically they are not stable, but they are increasing the surface area. So let's compare this increased surface area with Noyce-Whitney equation, which is like a standard equation for dissolution experiment. You can see the amount of drug released with the time is directly proportional to the area. So if you have API, which is converting into the molecular form, which has more area, and we can say dissolution is increasing. So this is simple correlation that we can do here. Now, coming on to the technology. Yes. So coming on to the kind of, amorphous solid dispersion, what kind of amorphous solid dispersion we have. Before discussing the type, let's discuss this graph, what we have. So you have drug in a crystalline form. You give energy to this drug crystal, they're going to become like amorphous. So overall, they are properly organized. Now this organization is gone and they are discrete molecules. They are still come close together, but they are not properly organized. So we can say that these are diamond, these become glass, which is not having any direction, and they are randomly distributed into the solid. So this is called amorphous. Now, once you give more energy to them, they're going to become individual molecules. So this is we call molecularly dispersed. So for this, you can see, if you want to break the crystal lattice, you need to give energy. And now, if we come on this part, you can see glass suspension, where you have a matrix. And in the glass suspension means you can already correlate with that, it having some suspension kind of molecule. It means some aggregation is there. So in the matrix, if you have drug molecule which is in crystalline form, and if you have a drug molecule which is in amorphous form, both are considered as a glass suspension. And one more part is that if they are molecularly dispersed, means we have given more energy to this molecule and they are properly distributed, or we can say properly dispersed in the matrix, we call glass solution. Now, if we compare this kind of technology with the matrix and can correlate with the type, so I will come on this part. Okay, so we have type 1 to type 3, which having matrix is more crystalline in nature. So this matrix which we are talking here is become more crystalline. Then these become this type 1 to type 3 type of amorphous solid dispersion. And if you correlate with this one, stability obviously crystalline, there is no thermodynamically stability issue, means they are more stable. But in case of amorphous and molecularly dispersed, the stability is kind of low, little bit lower. Now coming on to the crystalline glass suspension, amorphous glass suspension and glass solution. So whenever the glass word is we use, it means the matrix is amorphous form, and drug can be crystalline, amorphous and molecularly dispersed as we discuss in this graph. So our interest should be focused in the direction of amorphous and molecularly dispersed API. So amorphous matrix and molecularly dispersed API, that's how we can achieve maximum resolution. And if you see the stability issue, so amorphous and amorphous drug is kind of least stable, but amorphous and molecularly dispersed API, we will have highest dispersibility. Now, how we can achieve this highest dispersibility? So presence of some functional group, either on, like we can say, on the matrix or maybe drug, if they both will have some kind of functional group which can interact each other, they can form this kind of system. We'll discuss later once we come on that part. So let's focus on this. Here is amorphous glass suspension and solid glass solution. And what generally happens when you see this kind of matrix. So when you prepare amorphous glass suspension, you can see we have matrix. This is polymer, we can say. This is polymer and this is drug. And when you prepare them, you can see what happened, that slowly these amorphous drugs generally come close together, and then they started eventually come close together and they're going to form one kind of aggregate. So with time, this is kind of, we can see in this kind of problem, in this kind of system, that the drugs generally come close together and they form aggregate. And once they're going to form aggregate, their performance is going to indicate impact. We're going to discuss that one in next slide. Now coming on to the solid glassy solution, here what we have is, we have drug which is already interacted with our polymer chain, and because it is very well interacted, it means these molecules cannot move, they are locked, and that's what we are looking for. So once they are locked, now they can be more stable, and when you disperse into the water, as we discussed, this polymer will orient in different form. It's going to form kind of micellar structure, and the drug will be trapped over there, and then it will increase the overall dissolution rate. So this is what we can see in this kind of, difference between the two type of sets. I hope we convey this message to our students. Now we come on to the part where we want to discuss about the problem. So once you create this happy guy, means you have amorphous solid dispersion and molecularly dispersed drug, you can see the dissolution is really nice. With the time, the amount of drug dissolved is getting higher, and then it's going to reach to the 100%. So this is happy guy. Now you have one sad guy which has a matrix, and the drug is in crystalline form. What will happen? The drug may release, but because it has higher crystallinity, immediately it will crash out in the dissolution system. This generally happens in a storage condition also. So if your drug is not properly oriented in the matrix, or it is not molecularly dispersed, it is generally get crystallized out, which we were discussing in the previous slide. Now it becomes crystal, so it will release, but eventually it will crystallize it out. This is sometimes we observe, or sometimes we don't observe any dissolution. So all scenario is possible, but ultimately this unhappy guy makes overall drug unhappy and patient unhappy, because once you take this medicine, this API is not going to dissolve, and it's not going to dissolve means it's not going to get absorbed, and it's not going to show any therapeutic efficacy. So this is a major hurdle in overall amorphous solid dispersion we see. So stability is a big issue. Now let's discuss about some of the parameters, storage parameter, which generally affected by different property of API and matrix. So let's talk about TG of the matrix. So generally for this kind of system, we need a matrix. Matrix means polymer, which should have higher TG. So higher TG means, I think maybe many of students don't know about TG then, so let me clear about this part. TG means glass transition. So as we know that when you heat any matrix, what happens that slowly after heating, they at certain time point, they become like mobile because their molecules are started to lose overall bond and everything. And because of that, they become really flexible. And if they become flexible, then the API will get the chance to move. And because if they will get the chance to move, they're going to become crystalline form. So this is a major problem we see in a kind of low TG matrix. But if you have a high TG matrix, this polymer chain is not going to be flexible, and the API is going to be remain in the place where it's supposed to be. Now coming on to the next point is interaction of matrix and API. So this is a point which I was talking about, where you have a drug which have some functional group and matrix have some functional group. If they get the chance to interact, then it's much better. Sometimes, if you look, API and matrix have hydrogen bond. So this is very good. If you have hydrogen bond, they are more stable. Sometimes matrix and API forms some dipole kind of interaction or weak van der Waals interaction. They are weak. It means the drug will eventually get the chance to crystallize it out. So this is how pretty much we can correlate with the interaction of matrix and API. Now here in the third point, we are talking about compatibility of matrix and API. So it's not like chemical compatibility. We are talking about sometimes physical compatibility. Here what happened that API as such is not stable in the matrix, and then this is not a good system. Now coming on to one more part, which is we're going to discuss later, but I want to point it out here is sometimes when you prepare extrude from hot melt extrusion, then the cooling rate is important too. So if your cooling rate is smaller, like faster or it's slower, that also changes the drug mobility in the matrix. So if you are getting like hot melt extrude, which is really hot, and you take the time to cool it down, ultimately what will happen, because it is very hot, the polymer chain is more flexible, and API will get the chance to interact. So cooling rate is also one of the major issue which we generally see. Now dissolution parameter, I think maybe I'll not go in detail into this, but simple is that matrix dissolution rate, if your matrix is hydrophilic in nature, then immediately it will dissolve. Sometimes what happens that your drug will release faster. And where in this case, in the GIT tech, what we want is the precipitation rate must not be higher than the absorption rate for the drug molecule. This is pretty much we generally see. So if your absorption rate is faster than the precipitation rate, then it's better. So if your matrix is dissolving very fast, then what will happen? The API will going to come out. Eventually, you're going to see it's going to come out, but it's going to crash out because API having high tendency to crystallize it out. It will affect the overall performance. Now coming on to the one part, how we can prepare these amorphous solid dispersion. So as you can see, we have three major technology available in the market. One is hot melt extrusion, which is a solvent-free technology. So you don't need any solvent. This is a great advantage we can see in this hot melt extrusion. You don't need any solvent at all. So it's very easy. So solid in, solid out. This is a major advantage of the hot melt extrusion. But if you look the spray drying technology, it is also good technology, but you need some kind of solvent. For example, you need ethanol, methanol, which generally dissolve the drug and polymer, and then you spray them and then collect in one chamber. So this is called pretty much spray drying. So basically spray and drying. Now coming on to one more technology, which is I think maybe advantageous for developing countries like India, where many of the companies, I know that many of the companies are now hot melt extrusion. Before that, they were trying to do drug layering. When I was in India, like 2008, 2009, companies are trying to do more, drug polymer layering because they don't have facility to establish hot melt extrusion. So I work on this kind of technology also. It's very simple. You need to take non-sterile sheet. You dissolve your drug and polymer, and then you spray. And then what you're going to see is sometime the polymer will come on. It's like a more granulation from the, if you do the top spray. If you do the bottom spray, you can see more woods coating where individual seeds will get coated with the polymer and drug. So this is a difference between the top coating and bottom coating. And you might have, tangential coating also, which is generally also a kind of modification of this kind of technology. So we'll discuss today hot melt extrusion. I'm not going to waste the time, and let's start hot melt extrusion. So here in hot melt extrusion, this is overall view how this overall instrument looks like. I try to capture all kind of event which is taking place in the hot melt. So first we have upstream. So you have powder, you feed them, and you can feed the polymer. Sometimes company don't have two feeder. What they do is they mix the drug and polymer differently, and then they're gonna feed. So through the hopper, it's gonna go into the screw. So this is a yellow color is a screw where it generally start rotating. So screw can be twin screw, it can be single screw. So once they start moving, you can see there is a kind of a Lego we have. This Lego generally or screw generally guide the powder. So they start moving in this direction. And here we will increase the temperature. So temperature can be higher and depending on the need. So in the middle zone, generally we try to keep the temperature much higher so that the polymer will melt and the drug will dissolve. And here we want to see the less temperature. So once the purpose is solved, the drug is solubilized in the polymer, you cool it down. So we also have some kind of cooling liquid, which is also assembled in this hot melt extrusion. So chiller generally circulate the chilling liquid. So once you need certain temperature, the chilling liquid will move inside and it's going to cool down this whole chamber. So here is hot, but here is cool. And ultimately what will happen, it will extrude out from this hole. I think maybe for Indian people generally know how we generally prepare maybe kind of, we can say jalebi, or maybe we can say noodles, or so generally we know that how we generally, noodles prepare. So this is the same kind of instrument, but it looks fancy in terms of scientific, setup. How we generally create. So you squeeze the material and basically it's going to come out. So once it's going to come out, this molten polymer becomes solid. And now it's kind of hot. So you need cooling chamber. This is a cooling belt. Sometimes the roller also is kind of, chilled. So once they're going to move from here, it's going to become really solid. And then now you have granulator. So it's going to chop it down the overall extrude in pieces. Now you can see we have one milling. So this milling, what we're going to do is the small pieces will going to mill down and then it's going to pass through one sieve. Now again, it's going to convey it here and then you can see it's going to go to the V-bend blender. And once it's going to go to the V-bend blender, you can able to see, you can able to mix other excipient side, disintegrant you can mix, or maybe lubricant you can mix. And once you mix them, and then you can load into the tablet, and then you're going to get the tablet out from the tablet press. So pretty much you can see here solid in, solid out. This is the main concept of the hot melt extrusion. You can do this process continuously. So you don't need to stop this process anytime. Like in case of normal granulation process, what we generally do is you prepare granules, then you need to dry. Once you're going to go for dry, that's a limiting process. But in this step, you don't need to wait for anything. You just do the continuous, overall analysis as well as, manufacturing. Sometimes people generally put digital, we can say detector here, who going to monitor the ratio of drug and polymer so that you can do the quality check at the once the material is coming out from the extrude. So it's very easy to do it. It's not very complex. It looks very big instrument, but it's very easy to handle, and it's not having. We can say it's having less regulatory hurdles once you optimize because it doesn't have any solvent. You can monitor the overall quality parameter. Continuous manufacturing is possible. Now, one point here, which sometime initially when I start developing this hot melt, like product, I used to get sometime confused, or maybe whosoever is looking this one first time will also get confused. Like if you moving this material it may be possible that it will stuck here. But the answer is no, because you can see how this screw, if you look from this side, you can see how the screw is moving. So one screw will take the material here, and then the second screw will having less gap. So what it will do is to do the cleaning. So one extrude material or maybe screw will take the material, another will start cleaning, and then it will also taking the material. So they are moving in opposite direction. Sometimes they move in same direction too, but opposite direction will help in overall cleaning the screw. So once you've done this experiment, you open the instrument, you can see the screws are really clean, and you need to hardly clean it. And cleaning is like there is another validation we not discuss today. But let's move on to like, twin screw extrusion process and what are the things we generally need to, see. So they have different zone. So first is feeding zone. So pretty much is to convey the material. And you can see the pitch here is bigger and compared to here. So what will happen, the material will come here and then it's going to move. But as it's going to move inside this barrel, what will happen that the screw pitch is getting smaller, and because of that, the air will remove. Now, if the material will move here, you can see these are the screw. So how is it changed? So you can see here I'll come on this part. So you can see the screw pitch is bigger here, and the screw pitch is smaller. So bigger will move the material faster, but smaller will move the material slower. So ultimately, what will happen? The air will gonna come out, which I was talking about here. Now, here the screw design is different. So this is one screw, big screw, and you arrange these Legos on the top of this screw. So this motor will move, the whole screw will move, but because you have different kind of Legos, they're gonna move at different RPM or different style. Now you can see we have one kind of Lego which is like, you can see this is 30-degree angle screw, and here is 90-degree angle screw. So this generally affects the overall distribution or breaking down of particles. So which we're going to discuss in mixing zone. So let's focus on this part. So we have melting zone where you melt the polymer. Now, once the melted polymer move, then you have one more feeding zone. Sometimes people generally use in order to inject the liquid. So once you have polymer in liquid, and if you want to inject maybe some solubilizer, maybe in liquid form, you inject here. Now, once this material will move further, you can see the mixing zone. Mixing zone has two main purposes. One is dispersive and distributive. People will get sometimes confusion, like, what is this dispersive and distributive? So distributive means you have big molecules, which is like a brick, and you are breaking down, they are becoming a smaller particle. But they need to distribute it. Once you distribute them, then you need to disperse them into the matrix. So this is what we call here is disperse. So small molecule will disperse in the matrix so that we'll get more content uniformity. So that's the purpose of this screw R. Now then we have homogenization, and then we have degassing. So because we are using polymer and the drug generally have some kind of solvent, and polymer has some solvent which generally trap inside the structure, or sometimes they have moisture, or sometimes they have smaller volatile impurity. So what generally we do here is we put the vacuum so that this volatile impurity will be removed, and now we have discharging port. So you have dye, it will extrude the material, and you can make whatever you want to make it. Now let me show you one picture. So this is a overall same instrument. You can see the material is coming here and crashing down in the mixing zone where it's going to melt. So this is pretty much how this hot melt extruder works. And one more, we have a picture here. Yes, it's working. So you can see the screws are moving, but you can see here how this mixing zone or melting zone is moving. So you can see overall how this mixing zone is going to help in dispersive and distributing the material. I hope I convey this message clearly. And now moving on to how the extrusion look like. So this is coming out from Thermo Fisher HME Pharma 11 that we have in our lab. So you can see the extrude which is coming out. It's like noodles. It's clear noodles. And now once you chop it down, which I was talking about previously here, so you chop it down. So once you chop it down, you can see it become pellet. So extrude and pellet. How they looks like, just for your information. Now coming on to one part where we can say hot melt extrusion we have, and we have here different temperature. So what we can do in hot melt extrusion, we know that we need to hot and melt. It means we need to increase the temperature. So once you increase the temperature, what will happen? Energy requirement, because these are big instruments. So big instrument, if you operate at lower temperature, they need more energy to move the material. And if you will increase the temperature, you can increase the amount of material coming per rotation of a screw. So this is green zone, we can say. Here I can show you. This is a green zone. And this is not green zone, means here you are consuming too much of energy. So once you increase the temperature, you can see melting of polymer, and you can operate at more economical way. This is once you establish hot melt extrusion, sometimes people generally do optimization. I'll quickly go through this graph. Here we have same kind of information, but it converted into the volume. So amount of material, volume coming out per rotation in gram, and this is temperature. So now when your temperature is much lower, your polymer is not going to melt, then there is no drug is going to dissolve. It's like a dry coffee, or maybe we can say powder, coffee. There is no water, then you try to make sugar and coffee, it's not going to mix. So this is the same zone. So once you increase the temperature, polymer will melt, and your API will sometimes dissolve. And now if you will further increase the temperature, what will happen? The temperature will reach much higher limit, maybe more than 200 degrees centigrade. Some polymer are not stable, or some drug are not stable, so there is degradation limitation. So now we will know that we need to operate between this blue zone. Now one more thing, if you will start feeding more material in this condition, what will happen? Slowly you will go and increase the amount of material which is coming out per rotation is going to increase. But at some time, what will happen that you feed too much of material, the machine will choke, and we can say torque limited. Machine cannot operate. This is generally happen with the small instrument, but if you have a commercial instrument, this is not a problem. Now, sometimes it's volume limited, means machine is having capacity. You can feed the material whatever you want, but overall volume which is coming out is going to be constant. So you can do this kind of optimization. Just for simplification, I explained this one. And what will we get is, you can see sometimes people generally operate at this condition. So you can see overall amount of material of which is coming out is this much. But if you will optimize, you can increase the cost effectiveness. So with the same operation increase, you can get more amount of material which is coming out from the instrument. Another aspect is that sometimes people operate at much higher temperature. We don't need. And if you decrease the temperature, you can get the same amount of material with lower temperature, which will help in overall stability or impurity profile of the product. This is one more example which I would like to discuss. So this is we have done for VA64. The same experiment, you can see the solubility limit here, temperature limit here, and torque limit here, and then we have feed limit here. So this is pretty much for our VA64 polymer that we have done. Now I will not discuss too much in this direction. Let's move on to the matrix. So we talk about hot melt extrusion, and we talk about drug, we talk about matrix. Now how to prepare matrix or how to choose the matrix. So in BASF we have lots of good polymer. If you choose the middle one, these are the gold standard. There is no issue. Polyidon VA64 is the gold standard in the hot melt extrusion industry. So this is very good. And we have Soluplus, which is another solubilizing agent, and it will act as a matrix former also. So this is for stabilizing the API. Soluplus generally have a stabilizing effect as well as solubility enhancement effect also. Now this is the amplified lipid. Now if your API is soluble in more water, you can take Colycoat IR, Colycoat Protect, or Collidone or Povidone material. These are very good hydrophilic material, but the problem we need to be more careful because these are hydrophilic, means they love water. So once you water-loving matrix you're going to make, it is possible that they will, in stability, they will absorb the moisture. And once they will absorb the moisture, we know that their TG is going to change because water is a plasticizer, and we're going to see some kind of stability issue. So we need to be careful while choosing the hydrophilic surfactant. And now we have Colidone SR and Colicol MAE 100P. MAE 100P is a little bit difficult to work. Colidone SR is an excellent sustained release polymer. It's going to maintain the overall matrix throughout the release profile. So you make matrix and you want to see diffusion of drug up to like 24 hours, you can do with the Colidone SR. Sometimes people wanted to mix and match the release profile. So you can blend Colidone, which is on the top, with the Colidone SR, so that you can make pore former and you can create whatever matrix you are looking for. It's going to be very, very useful for formulator. Now coming on this, how the polymer looks like, which I was talking about. So once you increase the temperature, their viscosity is going to decrease. As we know that viscosity is lower, we can operate the instrument more efficiently. So this is how this overall polymer looks like. Colidone 30, you can see on higher side, it needs much higher temperature, but VA64 is excellent. You can see overall how the viscosity is decreasing is very good for HME work. Now coming on to one part that you have seen, like, we need to operate sometime higher temperature, maybe starting from for Colidone VA64, 155 to 200 degrees centigrade. But if you add the plasticizer, what they generally do is they interact with the Colidone VA64 or polymer, and they lower the overall operating temperature. So in VA64, you can see it decrease the temperature to 20 degrees centigrade, and sometime you can see more than 20 degrees centigrade decrease in the overall operating temperature. This is very important because if your API is sensitive, you can mix the plasticizer and then you can do the extrusion much easier. We have Colisol PEG grade polymer, which is in liquid form. We have Polyfor P grade, polymer grade, we can say, which are available. You can see P188, P407 we have. This is a very good plasticizer. Keep in mind that these are solubilizer also. Poloxamer has very well reported for solubilization purpose also. So pretty much you can get the two purpose. You can get plasticizer as well as solubilizer. Now we have micrograde available, which is very important. Once you suppose your Colidone VA64, your drug is having some special part density or special smaller particle size, you can pretty much choose the micrograde material. If you don't have, if you need regular grade, then you can go with the P188, no micro. So depending on the need, you can tailor the overall particle size also. So we have both kind of polymer available in our portfolio. Now coming on to the excipient size, you can see we have very good solubilizer, Colifor RH40, Colifor, which is a polysorbate, 20, 60, 80. We have Colifor EL and Colifor HS15. So these are very good materials to solubilize the API, because sometimes API goes inside the matrix, they interact with the polymer, and it's not going to come out. So if you have solubilizer, it will come out. Or sometimes you have drug which generally crystallize it out. You can use this solubilizer also during that time, because they have long chain which generally inhibit the crystallization. So I'm not going to go in the detail, but let's quickly discuss. So we have pre-testing we need to do. So in the pre-testing, we will establish the drug solubility in the polymer. Then we have solid dispersion. So we will do long-term stability study and physiochemical studies. Now we have processing conditions. So as we discussed, we need to establish the processing condition in order to make HME product, and then you can make final product depending on the API property. So immediate release, control release if it is very potent, and if it is having problem in the GIT, then you can make entry-coated. So depending on your need, you can tailor. I will not discuss too much. Let's quickly discuss how this material looks like because of the time constraint. So we have VA64, how it looks like. You can see at the 160 degrees centigrade. Colidone 12B, you can see 17 PF. This is parental grade material. And we have Soluplus, you can see. And pretty much if you have Colicoat IR and if you have Same with the Colicode Protect. Generally, they are good up to 160. If you will increase the temperature, they are not very good. In the case of P407, which has very low melting point, like around 60 degrees centigrade, they are melted. So extrusion, their chopping is difficult. So once the material is going to come out, you're not going to be able to cool right time, and then it's going to be having problem. Now this is very important for our student, because we don't have this instrument available in our lab, and then how we can do those kind of experiments and how we can establish the condition which is more suitable for HME. So this is for those students who want to do work in the lab. So what we need is simple thing like API. You take polymer, you take solvent, you dissolve it, and this generally this we call film casting method. So this dye generally run over the plate, glass plate. So and then you can add with the pipette also, and then you run over, you create one film which is less than 120 micron. And what you can do is you can store it room temperature for 30 minutes, and then in the vacuum, you can store it for 30 minutes again under 10 millibar at 50 degrees centigrade. And you can compare whether it's making glass solution or it is crystallizing out. So you can do this kind of experiment to understand what ratio you need. You can see the example, carbamazepine VA64, you can see the crystallinity present once you increase the amount of drug. At 20%, you can see the crystal. In solution form, like with solubilizer also, you can see the crystal. But in case of fenofibrate, without solubilizer, you can see crystal formation at 25 degrees centigrade. But fenofibrate with solubilizer, which is Soluplus, you can see much increase in the overall dissolution of fibrate. So this experiment you can do in your lab. It's very easy to do it. And you can establish the ratio which is going to work. And this is very much translated into the real world. You can see here, we told that 25% going to work. Then you can see here, this is extrude as such, then pellet with the drug, you can see they are becoming more opaque. But at 25 degrees centigrade, you can see it's like kind of cloudy. And then when you do the various characterization tool in the DSC with the 25%, we observe only 1 Tg, because this drug is having low melting point around 75 degrees centigrade. And if you're getting a TG means there is no melting. It's just shifting in the overall baseline. So here you obtain the TG in the DSC curve. DSC is differential scanning calorimetry. And then you can, if you observe TG, it means it's not in crystalline form. But when you increase the amount of drug, you can see melting. It means 30% it is becoming more crystal in nature. And the same experiment, if you do with the XRD, you can see the crystallinity of the drug. But this is a polymer itself, and then we have different level. At higher level, you can see there is a more crystal formation. But here, there is no crystal formation. And when you store them at 25 degrees centigrade, 75% RH, or maybe your stability condition, you can see the crystallinity change. If it is crystallinity changes, means the overall ratio is not established. And if it is stable, then we can say the overall formulation is stable. And this is pretty much we want to convey here is we have done the study using itraconazole, and we have solubilized the drug. And you can see here crystal drug is not getting absorbed, physical form is not getting absorbed, but HME is getting much higher bioavailability as compared to the free drug and physical form. So this is a great example in vivo, which we have done a study. So we can use 2 polymer. We have CA64, which is gold standard. This is a very important because we need more amount of polymer to dissolve the drug. And you can see the IID limit is 854 mg per day is approved. So you can create big tablet if you want. These are dust-free and excellent flowability and extrude up to 220 degrees centigrade. This is the advantage of VA64. Soluplus is great solubilizer, ideal for hot melt extrusion, and it's very unique property it has. So definitely I'll encourage those who are working in this field. They know that, but for students, if they want to work in this field, you can choose this polymer for your experiment. One more example, which is the last example I will say, is we have Soluplus. You can see you create the drug dissolving itraconazole 25%, how it looks like. You have Polydone SR, which is 50%. You can make the extrude. It means this is much faster dissolution. It is for IR. Polydon SR for sustained release. You can create sustained release. Now you can mix and match to match the release profile. This is also possible. Now coming on to one misconception, and I think this is the last slide before conclusion. So we can say common misconceptions are there. First is that API must be melted. It's not required because as we know that we dissolve the sugar in coffee. Sugar is in solid, but it dissolves in the coffee. In the same way, if your API having higher melting point and it's not dissolving, it's not melting, it's not a problem because your polymer will melt and it will dissolve the API. Now, people will think that this is a big instrument, overall resident time going to be higher. It means it will take time to come out and then drug will be destroyed or there may be more impurity generation, but it's false. You can see the experiment here. Once you run at optimum speed and you will optimize the parameter, API will come out like within seconds, so we can say within less than 5 minutes out. So with that temperature, it's not going to get the chance to degrade. Obviously, you cannot use thermolabile API, but it is pretty much a stable process. Process is too complex. Believe me, friend, this is not a very too complex process. It's very easy. It has been used in the plastic industry. We know that in our, in our daily market, if you go market, you can see hot melt extrusion product everywhere. Even but the tube which is coming for, supplying the water tube is also made with the hot melt extrusion. And if you see any film which is polythene is coming in market for the bag, or if you see the polythene roll, they are also made with this hot melt extrusion technology. So this is not a new technology. All patent which is filed in 1930 back to is already expired. And in plastic industry, this is very old kind of concept. But for pharma, it looks like new because we never thought in this direction. So sometime looking the things in a new angle going to help us to make a new kind of product. This is what we have discussed. So you have drug and polymer. You can make hot melt extrusion. You can add plasticizer. You can add solubilizer to enhance the overall processability. These are the parameters we already discussed, which we need to consider. I will not go into this. Now, summary part. So, poor soluble drug, we have a 3 pillar which we discussed: ASD, lipid-based drug delivery system, and drug polymer, and layering, which we can adopt, and these are trend in the market. So, this is highly in demand. So, if you're working in this direction and if you have expertise, definitely you are valuable to the industry. BASF has expertise, so if somebody is developing any kind of product and if they need any help from BASF, definitely we are ready to help you. You can join with us. We have Mumbai lab. Over there we have hot melt extrusion. We have over there technical representative. You can contact them. They're going to help you in overall development of their product, your product. These are the available portfolio. You can see for matrix pharma, we have different kind of matrix pharma. We have solubilizer. We have complex ingredient if you want to make a drug matrix complex, liquid solubilizer. And those who wanted to read more about this, you can download this free book right now available. It's kind of Bible for in hot melt extrusion. This is a very good book. You can download, very easy to understand, very simple language. Solubility, we have another book you can download from BASF website. This is for our who need literature. We have melt extrusion process, which is a design edited by the Nigel, who is a BASF employee, and he generally developed this book. Very important. I think it's available with the Spinger. It's not free. Now, if you need any help, we have experts all over the world. In India, you can see we have Raj Kiran, who's going to help you if you need any help. We have virtual pharma assistant. So not physical person. We have app also available on a website. It's free. For formulator, if you want to see what kind of dosage form you want to use, it's very good. We have excellence for quality and regulatory purpose. And if you want to procure anything, then we have My Product World app. So these are available. And with this, I will conclude my overall presentation. Thank you.
Thank you, Dr. Nitin. It is very nice, informative session. And you are nicely elaborates the material, how we can change the size of the size and shape of the material and enhance the viability as well as enhance the solubility. So very thanks to Nitin to elaborate the
welcome.
Science.
Yeah, welcome.
And there are some questions from the participants. So I am allowing to ask Professor Satish Bashar to ask your question. Satish, I allowed to you ask the question. Professor Satish, unmute yourself.
Yes,
Hello.
are you able to hear me?
Yeah.
Yes. Good evening to Dr. Nitin, Dr. Lindsay, and Dr. Power and other participants from Dhaka, Bangladesh. And in the meantime, Dr. Lindsay has already answered my first questions because I asked for the what is available for the amphotericin B and this anti-cancer drugs, because for the reductions of the toxicities to dose reductions is very important in that cases. So if it is very possible to increase the availability of the drug, definitely the size of the dose will be less. That is one concept for increase the bioavailability, so dose can be increased very less. That is one options. But these hot melt extrusions cannot be applied for intravenous preparation. That is one problem. That was satisfied by Dr. Lindsay. My second question is also supplemented by Dr. Lindsay, but also to Nitin. What is the benefit in terms of cost of the traditional tableting procedures than this hot melt extrusion procedure? So it will be a good thing, because this lecture is very, very academic and very, very interesting to the students and also for us also. Thank you, Dr. Nitin and Dr. Lindsay. I heard about Dr. Lindsay in the primary, in the middle of this talk. Thank you very much. To give me the opportunity to say something to the, Dr. Power and his team. Thank you very much, Dr. Nitin.
Yeah.
Yes, I will say, you rightly mentioned about, there is always this advantage of each technology. I will say, in case of a tablet, if you're going with the conventional method, you will get conventional kind of, drug delivery field, where you need to see like how the drug is releasing out. Sometimes it's going to not release properly. It's like depending on all patent issues. Now, if you're coming into the ASD product, you don't need any solvent. It is continuously manufactured, and it is possible. Not only one product, there are many products are available in market. You can see here the list, which I have not done because of the, we have some sign with the companies also, but pretty much we will not discuss in detail, but you can see Novartis, Janssen, Eli Lilly, Abbott, Pfizer, they launched the product, and this is give them evergreening kind of technology, because once you launch this product, somebody want to manufacture, they need to manufacture with hot melt extrusion, because it gives some unique advantage in terms of increasing the solubility, and you can reduce the dose, and you can reduce the toxicity, you can make the product stable at the room temperature. So there are many advantages of hot melt extrusion, and definitely I think maybe it generally gives different kind of edge to the, not only innovator companies, but for the generic companies also, where once you make this one, you're going to be out of the, you're going to look for different, and you're going to be able to increase the overall viability, and your product is more superior. So definitely I will say there are, we can say the technology differences, but obviously hot melt extrusion is much better technology.
Thank you, Dr. Nitin, for the answering. And I think it is more validated, this method, when it is established in a top-level companies, as well as in the innovator. In Bangladesh, there are some companies, top-level companies, they are contributing a lot. And so far I know, they have an intention to use such kinds of technologies in their industry, because this method is very, very validated. Batch to batch variation is very less in this case, because it's a continuous process. no interruptions,
Yes.
and this person involvement is very less. So,
Yes.
so the product output and quality is very high standards. So, so it is a very interesting. That's why I am very much happy to join in this lecture today, and thank you very much
Yes.
to supplement us. About
Thank
this.
you. Thank you.
Thank you.
Thank you for joining.
Thank you, sir. Thank you for joining.
Thank you, Dr. Pawar. Thank you very much. Very, very good academic talk.
Thank you, sir.
Thank you. Thank you.
So,
Thank
Nitin,
you.
there is one question from Kashyap Nagarpia. How much efficient this technology for sterile formulations?
Okay, this is very good question. And then if you know, medical tubes which are coming in market, where there is one tube which has like different holes, maybe 3 holes or 1 holes. These are, there's medical product also generally work by this hot melt extrusion. So sterile product, you can make it. The cooling is the one biggest issue over there, because in cooling, generally you need to dip the overall polymer, which is molten. Sometimes they use water. But in case of sterile product, you cannot do that one. You need a special kind of cooling system. That is a one drawback over there, but you can make the product, sterile product also with this one, not for injection, but for medical devices.
Okay. Now I would like to request our panelists to contribute your views on the talk of the Nitin, Dr. Nitin. Dr. Akash?
Yeah. Hi. So good evening all. So it was a very nice presentation. Actually, BASF is doing extraordinary work in the field of solubilization and biology enhancement. I think from many years they are in the field of development and commercialization of these novel excipients, whether it is a polyphore or it is a polysol. And today Dr. Nitin has nicely demonstrated the principles and mechanisms of this excipient for solubility and stability enhancement of API in a very simplified manner, actually, I can say. Frankly speaking, I also got some new understanding today in this discussion. But just to, from the part of my, some of the curiosity, I just want to understand,
Yes.
Especially from the regulatory perspective, like what are the expectations of a regulatory agencies like FDA or EMA on usage of these novel excipients for successful approval of a finished product, and whether these agencies need any specific characterization of these polymers along with a finished product?
Can I ask her? Yeah. So maybe, thank you, Akash. Akash is my classmate, I know. So Akash, for this one, actually, for this question, I think maybe is very rightly, question is very good. And you always worried about, what will happen if the polymer is going inside the hot metal extrusion and how it's going to come out. We have support for that. We have done lots of study. These polymer, which we have mentioned, is stable at the high temperature. There is no degradation. We have done the GCMS analysis. There is no fragmentation. There is no impurity generation. These are rock star, polymer. So you blend with your drug, which I mentioned that the misconception is that they degrade, but they're not going to degrade. It's very easy to operate, and you can immediately cool down the overall extrude. It's very good. It's very good technology, and I will say it's getting popular in India also. I know that many companies has HM instrument, and we are developing the product with them. So I know that.
Okay. So from the regulatory agencies side also, they are very much familiar with these type of excipients. So from regulatory filing perspective,
Yes.
there are no major hurdles.
No. There is no major hurdles, which I was telling you that you can see the product which is already approved. So many products are approved. And I was telling you about VA64, it is already approved in one product, many product, but you can see the higher D limit is 854. So it's regulatory approved product. Soluplus is still in a stage where we are trying to get approval in USA, but it's already approved in Europe, some of the, for new products and some of the other areas. But now in USA, still we are working in this direction.
Okay,
Yes.
so I think we had a lot of technical discussion today from last 1.5 hours, and Nitin is telling a lot of things which is very much technical for all the. I don't know how much it was easy for the students, which are the majority of our students, who are possibly the pharmacy students, how they take up all those things. but from something which could be more important to these students, like do you want to convey a message to them regarding their studies or their career or their aspirations, especially in the tough time of the current scenario, that how they can, because I know, as you told that we are the classmates, so I know you from last 20 years and I've seen you growing from basic stage to this level actually. And so maybe you could be a right person to aspire the students also, maybe who are listening these conversations, how they can take up the things, they can take up the opportunities to the next level.
Definitely, and I can answer this one in English or Hindi also, but I'll prefer in Hindi. So, you know, for students, if you are, so now switching the gears in Hindi.
Yeah.
तो जरूरी है कि हाँ, इस समय सब जगह पे क्या हो रहा है कि देख रहे हो कि कोविड-19 चल रहा है और ओवरऑल कितनी डिफिकल्टीज हो रही है, लेकिन डेफिनेटली स्टूडेंट्स को अपना मॉरल हमेशा हाई रखना चाहिए. और यू कैन सी लाइक यू नो फार्मेसी इज़ अ वेरी गुड फील्ड, जहाँ से मैंने बहुत कुछ सीखा है, बहुत कुछ लर्न किया है. मैं वहाँ से मतलब यहाँ तक पहुँचा हूँ. मैं भी वही सागर का रहने वाला हूँ, एमपी का, तो वहाँ से मैं यहाँ तक पहुँच पाया हूँ बस ओनली फार्मेसी की मदद से. तो डेफिनेटली फार्मेसी इज़ अ वेरी गुड फील्ड. और मैं ये भी कहना चाहता हूँ कि जो आप यहाँ पे लर्न कर रहे हैं उसको ज़रूर याद रखें अपने, मतलब कहीं ना कहीं पे आपको वो काम आएगी. कोई भी स्टडी करते हैं इस समय, अगर लॉकडाउन है तो ज़रूरी नहीं कि आप लॉकडाउन है, पर यू हैव ब्रेन यू कैन यूज़ एंड लर्न मैनी थिंग्स विच इज़ अवेलेबल इन लिटरेचर, यू कैन गो थ्रू दैट और आपको बहुत कुछ सीखने मिलेगा उससे. और उसको याद रखो हमेशा लाइफ में और आप कहीं ना कहीं इम्प्लीमेंट करोगे उसको. सो इट्स वेरी गुड. और ट्राई टू बी फोकस इन योर ओवरऑल करियर. In my career, I was focused in my this drug delivery field, and I am still in that drug delivery field, so that I can perform better. तो यही है मेरा ओवरऑल संदेश. पर लर्न हमेशा नई नई चीज़ें करो, डाइवर्सिटी होना चाहिए. जैसे कि आप ये देखो ये टेक्नोलॉजी 1930 में बनी थी प्लास्टिक इंडस्ट्री में, और फार्मेसी तक आते आते 1990 में प्रोडक्ट लॉन्च होना स्टार्ट हुए. तो कितनी पुरानी टेक्नोलॉजी है, कहीं दूसरी फील्ड में है, लेकिन अपने लिए तो नई हो गई. तो वैसे ही है कि भाई अगर आप एक फील्ड की टेक्नोलॉजी को दूसरी फील्ड की टेक्नोलॉजी से मर्ज करोगे तो आपको बहुत कुछ सीखने मिलेगा. वैसे प्लास्टिक इंडस्ट्री से अपन कुछ सीख सकते हैं, कितनी अच्छे से उन्होंने प्रोडक्ट बनाया, कितना अच्छे से किया और यहाँ पे अपन ने लॉन्च कर दिया. तो वही है कि भाई टेक्नोलॉजी अवेलेबल है बट इन सम डिफरेंट फील्ड, सम डिफरेंट एरिया, एंड interdisciplinary, you know, if you can do more exchange of knowledge, it's going to be much better. And I think maybe this platform suitable for that. Am I correct, Dr. Pawar? Your presentation in this interdisciplinary,
Today is with us also Dr. M.S. Shuddis from Amrita University. They also focus on to the topic. Yeah, Sudhish.
Okay. First of all, thank you. Yeah. Can you hear me?
Yeah.
Yes, you can hear, Sudhish.
Yeah. First of all, thank you to Dr. Nitin and Dr. Lindsay. And Dr. Nitin happens to be my junior, and thank you for this insightful talk. I'm really happy that I could witness your talk. My question is regarding stability of these ESD formulations. Actually, as I know, there are two parts to this stability issue. One is this solid state stability, and other is the stability during dissolution of these ESDs as solid dispersions. What do you think, whether this solid state stability is more challenging issue, or whether the dissolution part of this ESD is the more challenging issue as far as the stability of the formulation is concerned?
I think maybe both are very important. We cannot say that, you know, which one is more important, which one is not important. I think both are important. Ultimately, we need to see therapeutic efficacy. So in both cases, in one case, the product is not stable and the storage condition is not good. So ultimately, we cannot see the efficacy. But in other case, like if it is getting precipitated out in the GIT tract, then I think we can say that that is not going to work. So ultimately, being as a formulator, I will say both are very important. And we should have to focus in both the direction. Sometimes, you know, with HME instruments, HME technology, if your matrix is dissolving very fast, what will happen? The drug will going to come out in the GIT tract, and immediately it will get precipitated out. For those kind of, you know, issues, we can tackle with including crystallization inhibitor. So we have many crystallization inhibitor available in our portfolio. You can take those crystallization inhibitor and then you can increase the overall bioavailability. So we have tools to do it, and we can take the right tools and make right product and a stable product which is going to perform in dissolution too. So this is overall our, I think maybe with motive to move forward. I think that's going to be our strategy. Thank you, Sudesh.
One more thing is that this dissolution test, whatever you do for ASDs, is it under sink condition or non-sink absorptive condition? What are the conditions for dissolution studies?
We generally, FDA has recommended for many products, but if it is something new, if you are working, then definitely we need to maintain the sink condition, which is the first requirement in any dissolution. So first of all, we need to maintain the sink condition, and then we need to do dissolution studies. Then we need to do in vivo studies. Then we need to establish in vitro and in vivo condition, which is more suitable for, sometimes you take some percentage of surfactant, and you're going to see the release maybe 2 hour 100%. But when you put into the GIT tract, you're not going to see like that kind of absorption. So we need to again tune the dissolution media in order to get suitable condition so that you can maintain in vitro in vivo correlation kind of thing, and that's how we can establish the dissolution condition. But you rightly mentioned about fast steady state and fade state media. We can definitely try, and that's one of the approach.
Yeah, really, thank you. It was really insightful of you. Thank you, Nitin and Dr. Linde, for a good presentation. Thank you so much. Yeah, over to you, Dr. Paul.
Yeah. So there is one more panelist, as I see it in the panelist, as Praveen Kumar. Have you any question, Praveen Kumar? Praveen Kumar. Hello. Okay, thanks to all of you to connect with us and focus on to the method which is related to the change of material named hot melt extrusion process. So thanks to all. In the last session, I would like to call our colleague Dhote, Vinod Dhote, to
Thank you very much, sir. Sir, it is my pleasure to convey my gratitude to today's chief guest, Dr. Lindsay Johnson, for her and informational talk to the participant. Thank you very much, ma'am. Next in this series, I would like to thank our resource person, Dr. Nitin Swarnakar, sir, for a detailed talk on the aspects of the solubility enhancement through HME method. Next, in the continuation, I would like to thank our management team for their support and guidance. Thank you very much, sir. Respected Shyam Rathore, sir, for his continuous support and presence during the this online lecture series. Next, I would like to thank Dr. M.S. Sudhi, sir, Dr. Akash Chaurasia, sir. As a panelist, they are present in this lecture today. Thank you very much, sir. Most importantly, I would like to thank all the participants from India and abroad and the viewers on the YouTube Live and Facebook Live for their presence in this event. Thank you very much. And most importantly, convey my thanks to convener of this event, Dr. R.S. Pawar, sir, Institute of Pharmacy. Thank you very much, sir. Thanks to all of you. Have a good day. Thank you.
Okay, thank you. Very nice.
Thank you, Dr. Pawar. Yes.
Yeah, very nice meeting, and it's like a small Sagar alumni. Yes. Thank you. Now I am going to end the session.
Yes.
Okay. Thank you.
Thank you, guys. Thank you, Dr. Pawar.
Thank you.
Bye.
Bye-bye.